All right, ladies and gentlemen, for today's presentation, we're going to learn about enzymes. Enzymes are going to be talked about here when we're talking about carbohydrates and lipids and proteins and nucleic acids, because enzymes are a very special type of macromolecule. Enzymes are a protein, all right? And you can see here from the screen that proteins are polymers, which we call polypeptides, and they are made of monomers, which are known as amino acids. We mentioned earlier that in a previous presentation that there are 20 different amino acids and they join together in long chains. And those long chains actually create a varying structure, even though the chain folds up into itself. And those structures change depending on the amino acids and what order they're in. And those structures give the protein a specific function. Now, enzymes are one specific function. All right. So the polypeptide unit of amino acids is the enzyme's structure. And if you recall, structure equals function. And so this structure will give us a function that speeds up chemical reactions. So let's talk about that for a second. All living things have lots of chemical reactions. Even the simplest of bacteria have a multitude of reactions that are required for it to be alive. You have thousands, if not hundreds of thousands of chemical reactions all happening at once in cells throughout your body. And they have to happen quickly. The thing is, they're just not going to happen fast enough on their own. So we have a special type of chemical known as a catalyst. Now, catalysts are any chemical that speeds up a chemical reaction. And there's an important addition here: without being changed. So what that means is that when I have a catalyst in a chemical reaction, I start the reaction with the catalyst, I end the reaction with the same amount of catalyst. And that's really important because then I can reuse catalyst over and over and over again. Well, enzymes, which are proteins, they're polypeptides, they are made of strings of amino acids, they are biological catalysts. So they are the catalysts for life. Now, the most important thing here to understand is that without enzymes, chemical reactions in living things do not happen fast enough for life to exist. Again, these chemical reactions will happen, they can happen, they do happen, but without enzymes, they cannot happen fast enough for life to exist. So let's talk a little bit about how it is that catalysts speed up chemical reactions. So what you can see right here is a little graph. All right. Now this graph, it's very common. You can see over here, we have the energy going into a system. And this bottom line here is time. So what's happening is, here is what I start my reaction with. I start my reaction with something called reactants. And my reactants have a certain amount of energy. Now what happens is, I need to give my reactants this much energy. If I don't, my reaction never happens. Think about it this way. When a match lights, it burns. The match burns until there's nothing left. But matches don't just light on their own. You have to strike them. Striking the match is adding activation energy. And once you get here, which is you striking the match just burns until it reaches the bottom of its energy. And that's the thing about catalysts. See this? Here, let me get rid of this for a second. See this green dotted line right here? See how it's lower than the red line? That's because when you have a catalyst, you don't need this much energy to get the match to burn. You only need this much. And so catalysts allow your body to make reactions happen quicker because you can use the energy you have to get more reactions started. All right? So that's very important. We say that catalysts or enzymes speed up chemical reactions? They do it by lowering activation energy. Okay? Now over here, earlier we circled reactants, which are the things that you start a chemical reaction with. Well, the reactants are what the enzyme acts upon. And so they have a special name when we're talking about enzymes, called the substrate. Now this is important. This was actually in your reading. You have thousands of different enzymes inside your body. All living things have multiple enzymes because you have a different enzyme for each substrate. And the reason for that is because enzymes are a structure, and that structure has a specific function. And that function is to only work on one substrate. Let's talk a little bit about why. So we like to say, this is a summary of what we just said, is that enzyme structure is substrate specific. And you can see written here in black, thousands of enzymes are present in complex living things. So you basically have one enzyme for each substrate. And to sort of explain why, I want you to take a look at this diagram. This diagram is known as the lock and key model of enzyme function. It's a very common model. In fact, it's generally the accepted understanding of how enzymes work. We have a lot of evidence that backs this up. So right here you can see the green structure, and that is our substrate. And you can see how this curved edge right here can only fit here. This sharp edge can only fit here. And what you'll see is, see how they fit in and they kind of change the shape of the enzyme a little bit? And then what the enzyme does is, the enzyme allows the chemical reaction that breaks these two in half to happen. And that's why you have two different substances right here. And then once they become two substances, they no longer fit. And so they come out. Notice how the enzyme here at the end is the same as the enzyme here at the beginning. Because enzymes are unchanged. But every enzyme only works on one substrate. So if I had a different shape there, say for instance, I had this. All right, so even though here it's just a flat bottom instead of a round bottom, it still won't work. I would need a different enzyme for that substrate. All right, that's what we mean when we say enzymes are substrate specific. Now you notice here I have this word circled in red that says active site. And our little arrow says enzyme part that acts on the substrate. So enzymes are generally big molecules. They're long chains. They have hundreds of amino acids. And so they have like different sections on them. And the most important section on an enzyme is known as the active site. And the active site is what does the enzyme's job. Okay? So this is what does the enzyme job. It is at the active site that the substrate actually fits in. It's at the active site that the chemical reaction actually gets sped up, that the activation energy actually gets lowered. The active site in an enzyme is where all the action happens. All right? So, one last thing about enzymes. So enzymes are proteins. And structure equals function. Remember that. It's one of the most important things you're ever going to hear in biology. Structure equals function. Well, the structure of proteins is very fragile. Very minor changes can screw up the structure of a protein. And if you don't have the right structure, you don't have the right function. When that comes to enzymes, it means they don't work. They won't lower the energy of activation, which means they won't speed up chemical reactions. And the chemical reactions then don't happen fast enough. We mentioned when we were discussing the characteristics of life that one of the main characteristics of life is an attempt to maintain homeostasis. And homeostasis means a constant internal environment. Okay? Well, and in fact, some of the most important parts about maintaining homeostasis are about keeping protein structure intact. If your internal environment is not right, your proteins get all misshapen. They lose their structure, which means they lose their function. A couple of good examples. Your body temperature. Different organisms run at different body temperatures. We all should know human beings run at about 37 degrees Celsius or 98.6 degrees Fahrenheit. If that temperature gets too high, 40 degrees Celsius, 45 degrees Celsius, your enzymes and other proteins begin to fall apart. They lose their structure, which means they no longer function. If your pH, remember how acidic or basic you are, gets too high, your body gets too basic, your enzymes fall apart. If your pH gets too low, too acidic, your enzymes fall apart, which means that your proteins lose their structure, which means they lose their function. When enzymes or proteins lose their structure, we say that they denature. Great example of this in everyday life. You cook red meat. Take red meat out of the package. It's soft, it's squishy, it's red, it's pink, and then you cook it. And remember, when you cook it, you're cooking it with heat, which means you're making the temperature too high. All of a sudden, the red meat isn't red anymore. Now it's brown. The red meat isn't soft anymore. It's soft, but it's still almost like a little rubbery. It's not as squishy. It's got like solidness. It's a solidness to it. That's because the proteins, red meat is protein, right? That's why you eat it. The proteins in the red meat, the heat caused them to denature. All of a sudden, they have a different structure. They have a different structure. They have different functions. They feel different. They look different. They even taste different. And if it applies to red meat when you cook it to eat, it also applies to the enzymes and the proteins that are inside any living organism that are helping it function. All right?